Okay, welcome to chapter one of biochemistry. So, a little background, I'll be doing these lectures for each section. I've posted office hours in the syllabus and also a link in Canvas. And so I'll be available 8:30 to 10 each morning, Monday, Tuesday, Wednesday, Thursday, to answer questions. But the main delivery of material will be through these lectures that I'll be posting on YouTube. And we'll proceed as outlined in the syllabus. We'll have some exams, we'll have some worksheets, and a lot of this we'll just build up to. So we're going to start off easy with chapter one. If you read it, there's a lot of little pieces in there, but not a lot of meat to digest. It will focus on things that you should know from your biology and past chemistry classes, and I'll try to hit those. But in the end, there's not a lot of what I would say biochemistry. It's more framing what we'll talk about. Chapter two, we'll get a little bit more into biochemistry where we focus on water. Again, chapter two will have a lot of review of general chemistry. And then finally, we'll get into biochemistry hardcore in chapter three. So make sure you understand chapter one well. Like I mentioned, there really shouldn't be anything new in here if you've had organic chemistry, general chemistry, and biology. So we're going to introduce you to the chemistry of life. I'll start off each chapter is by covering or giving the outline. This is material that can be found in your book, but I think it's good to understand the key points that we'll hit. So I won't spend a lot of time on this, but we've got three sections to cover: the origin of life, cellular architecture, and thermodynamics. So in this first lecture, we're going to cover those first two: the origin of life and cellular architecture. And then we have these key concepts. Again, these are found in your book, and you also notice that they are listed in the study guide that is posted. So these are the key things that we want to talk about. It'll put and frame our conversation for this first section. We'll talk about biological molecules are constructed from a limited number of elements. We'll talk about functional groups, again, a review of general chemistry information. Just talk a little bit about chemical evolution and how we may have got complex biological systems from simple compounds, and then bring up this idea of self-replicating molecules. And so we have a picture of a cell down here at the bottom that's been stained. And that's really just to help you understand and visualize the complexity of a living system. These aren't molecules even, these are larger structures. So cells are very complex. But this complexity actually comes from just a few elements. So if we go and want to classify living matter, it tends to have a high degree of complexity and organization. We extract and transform energy to create and maintain structures and to do work. So often individual components interact in a coordinated and a dynamic way. So living matter tends to be able to sense its environment and respond to the changes around it. If it wasn't able to respond to changes, it would quickly pass. And a key for living matter is as a capacity for fairly precise self-replication. So it can take information and pass it on to a new generation. And this is all accomplished by using a variety of elements, with the majority of life being made up from carbon, nitrogen, oxygen, and hydrogen. But really, all life contains what I was taught: CHONPS, CHONPS, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Those are the elements of life. And so those are incorporated into much larger and much more complex molecules. But you can see here, if you took a human and dried them out and analyzed the elements, you can see that most of it would be carbon, nitrogen, oxygen, and hydrogen. We have calcium, phosphorus, that's found in our bones, potassium, sulfur, chlorine, sodium, magnesium. But that's not all the elements. In fact, this is really interesting. So if we take Dave and we do the same thing, we dry him out and then figure out all the elements that are in them, what we see is that we have tons of elements in us, such as gold, zirconium, niobium, silver, antimony. And what's interesting is rubidium. That's the most abundant element in the body that has no known biological role. So if we took Dave, let's say that he weighs 170 kilograms, we would find 0.68 grams of rubidium in him. But we're not sure what that's for. And then we actually have vanadium. It's the least abundant, so it's found inside of us. And again, in 170 kilograms, 0.11 milligrams of vanadium. But again, not known. So maybe this is just incorporated into our body through our diets. But a lot of these are actually useful. Selenium's actually used biologically. And if you're deficient in some of these, you tend to get interesting diseases. So even though we're composed majority of just a few elements, our body actually uses quite a few of those on the periodic table to get work done. So the question is, if we're talking about biochemistry, well, where did life come from? It's thought that life arose on Earth in between 3.5 and 3.8 billion years ago. So they think they have some fossil evidence of life in some really old rocks. I believe these come out of Australia. And so they can date the rocks. They know that they haven't been really perturbed since their creation. And they found this interesting fossil. And so you can see that they also do a cartoon sketch of it. And so it's thought to be kind of a filamentous bacteria. And so by dating the rock, they've come up with an age of 3.5 billion years. So somewhere between 3.5, 3.8 billion years ago, self-replicating life. But to be self-replicating, you need to be able to transfer that information from one organism to the next. And so how is that information stored? It could have been carbohydrates, again, which we'll talk about a lot more in depth, DNA, could have been RNA, could have been proteins. So what's interesting about all those above, they're found in all life, and they're polymers. They're made up of small repeating monomers. So even entities on the edge of what we define as living contain these macromolecules. So viruses that you're hearing lots about, they contain these molecules. They contain carbohydrates, maybe not both DNA and RNA, but viruses will contain either DNA or RNA and proteins. Now prions, they're not necessarily contain all of those, but they are small proteins that actually can convey information from one molecule to another. So we're going to be talking about living organisms, those that can have offspring and transfer that information. Viruses can't self-replicate. They need another living system. So they're kind of on the edge of what we consider living, but they do contain all these biomolecules. And so how do scientists think life started? So we know that life's on Earth, trying to figure out where it came from. And there's been lots of thought about this. And what we've got to do is make the connection between what we think early life looked like. Well, sorry, not early life, early Earth looked like, and what modern day life looks like. So early Earth's atmosphere probably had water, definitely nitrogen, carbon dioxide, methane, ammonia, hydrogen sulfide, maybe carbon monoxide. There's actually lots of different thoughts about what actually composed the early Earth's atmosphere. It was thought that there's not much O2 out there. O2 was eventually became abundant due to plant life, which we'll talk about later. But somehow we got to get from these small molecules, these gases, and get to larger molecules, because we know that living things are made out of polymers. And so here is a little scheme to show you what a polymer is. A polymer is composed of monomers. Monomers are individual units, and when we link them together into very long chains, then we refer to it as a polymer or many mers. Okay, having some technical difficulties. I think we got it worked out. Anyway, so these monomers, small molecules, are linked into these big, long polymers. So for life to start, these compounds must first have formed at the monomer level and then eventually into the polymer level. So there's some interesting experiments. One that's probably best known is the Miller-Urey reaction. What they did is they subjected a mixture of water, methane, ammonia, and hydrogen to an electric discharge for about a week. So you can see the apparatus there. And actually, it looks like in this example, they had hydrogen sulfide. There's been lots of variations on this where they changed the amounts and types of gas. But they put some electrodes in there and create a spark, which was thought to be similar to lightning, which again was thought to be abundant in early Earth's life, and kept it at a fairly high temperature, 80 degrees Celsius, and then just condensed what fell out and then trapped it in a liquid. So they ran it for about a week, and the result was they found some amino acids and other biologically significant molecules. In fact, recently they've gone back and checked those samples and found over 20 different amino acids and other building blocks for maybe not this experiment, but in other experiments they've been able to create nucleotides and again, just a bunch of biologically significant molecules just by combining very simple precursors. Again, since no one was around in early Earth to make these observations, these are experiments that say how things could happen. Other people have suggested that the molecules weren't made in the atmosphere by lightning. They were actually made on the sea floor. So these are referred to as black smokers. And there's other types of hydrothermal vents. They release hydrogen sulfide and other metal sulfides. And you've got organic or the precursors to organic compounds dissolved. And under these high temperatures and in this environment, it's thought that biological molecules could have been made. So some very interesting experiments that give us a hint of how biological molecules could have been made. But of course, no one's been able to replicate and make life de novo from just these simple molecules. One of the things that we know that needed to happen for life to evolve was, again, the formation of polymers for small molecules to get together and make larger molecules. And so a reaction that's quite common in biochemistry is condensation reactions. And that is when we bring two molecules together, such as this example here, we're bringing a carboxylic acid and amine together, and you get loss of water with the formation of a new functional group, an amide, or in terms of biochemistry, a peptide bond. So we see quite frequently these condensation reactions. Again, condensation reactions are just the combining of two molecules with the loss of a small molecule, typically water. One of the issues in trying to justify how life came about by the condensing of these molecules is the reverse reaction tends to be more favorable, especially under basic or acidic conditions, the hydrolysis or the breaking apart of those molecules. As well as this isn't a very entropically favored reaction. You're actually taking two molecules and you're making big, long molecules. So even though you do have the loss of water, you are getting more organized by making these big polymers. And so in order for this to be the process that helped pave the way to life, the forward direction or the condensation rate must have been much faster than the hydrolysis rate. So as I just mentioned, we can make amide bonds through condensation reactions between a carboxylic acid and an amine. And just to remember, here are a bunch of the other functional groups that we learned in organic chemistry. Now, some of them that we only briefly talked about were thiols and disulfides, thioesters, and we really didn't talk about these phosphate esters and phosphoroanhydrides or mixed anhydrides. So these are going to become a lot more important in biochemistry. There's an interesting one up here. This is a guanidinium. That may be familiar to those of you that memorize your amino acids. That's the arginine side chain. So most of the functional groups that we talked about in organic chemistry, we will see again. And these functional groups are what links these small molecules into, again, these much larger molecules that eventually allowed for life to exist. So here's just an example of a very simple amino acid, glycine. That was one of the amino acids that they found in the Miller-Urey experiment. They could make glycine in there. And if glycine undergoes a condensation, then you get polyglycine or a big long chain of glycine. And so if we can justify that we get a high enough concentration of these monomers that they start to react together to make these polymers, well, the question is, well, how do they replicate? How do we make new molecules based on the old molecules? Because in order for life to exist, we have to transfer information from one generation to another. So it was thought that maybe these macromolecules actually have complementary arrangements. So something on one of the molecules would be attracted to a complementary type of interaction on another macromolecule, and that would help that other molecule form. It would also lead to greater complexity, and greater complexity would lead to greater functional possibilities. Again, we're just kind of skimming over this for a couple of reasons. First, people don't really know, but we just want to give you kind of get you thinking about, well, if we start looking at life, where did it come from? What are all the complexities that had to be solved to actually give rise to organisms? So if we look at it, first of all, we had to make macromolecules, and then those macromolecules had to be able to replicate other very similar macromolecules. And it was thought that maybe this just didn't occur in solution, that maybe it occurred on clay surfaces. So clays actually have a very organized structure. And so the clays could have acted as a template or a catalyst and made a bunch of very similar molecules. If you think about it, we've got a very long time for these reactions. You thought watching a reflux in organic chemistry for an hour and a half was long. You know, we had billions and billions of years for these things to start working. And a billion years is a long time. So, got some more technical difficulties. Be right back. I don't know what it is today, but we'll keep plugging along. So, again, these macromolecules that we see in all living things exist of proteins, where the monomers are amino acid, nucleic acid, where the monomers are nucleotides, and polysaccharides, so complex carbohydrates, where the monomers are monosaccharides. Again, we're going to have chapters on each one of these. Right now, we just want to understand kind of their complexity and make the connection between early Earth and simple molecules to these complex. And again, just try to understand how this could have arisen. So proteins actually are connected by these amide bonds right here, and they vary in their side chains. Nucleic acids are connected by sugar phosphate linkages, and they vary in their side chains by the heterocyclic base off the side, the pyrimidines or the purines off there. And then carbohydrates have these acetal linkages right there, and they don't vary much off their backbone. They tend to have alcohols. Every so often you'll actually see an amino sugar here, either just an amino sugar or an acyl amino sugar. So the polysaccharides actually probably have the least amount of decoration off of them as far as off of the sugar. The polysaccharides can actually form some of the more complex structures because of the number of sugars and the different types of branching. Proteins tend not to be branched. There are some cases where they're branched, but they tend not to be branched. And they just have side chains. The nucleic acids aren't branched. They just have the heterocyclic bases. But carbohydrates can be highly branched with a lot of variation in there. Okay, so if we start thinking about how we got these polymers to actually go and self-replicate, is thought that maybe clay particles catalyze the condensation reactions. And so you get concentrated small molecules, and you get them in kind of a high concentration on this clay surface, and it's in proximity to some other macromolecules. Then they could kind of template. And so you could see that these would match up here. That would bring them into close proximity. And then you have something like a surface that maybe has acidic or basic residues that would then allow those bonds to form. And now you've got two molecules that could also fold up on itself and be complementary. And then you could imagine that this continues on and you make more and more complex. So it's thought that initially the biomolecules arose through complementary means, meaning you made one molecule and then it helped direct a complementary molecule. And now since you have twins molecules, and then those would continue to make very complementary molecules. Okay, so we'll leave behind a lot of that talk about evolution and how life actually evolved. We just need to understand that life is very complex. And to get that complexity, there were lots of things that had to be solved, such as making complex molecules and getting those molecules to replicate so that the information could be transferred from one molecule to another. And again, this happens with very few, kind of a small subset of the periodic table. And you can see here kind of the ranking of those that are found in biological systems. Again, carbon, hydrogen, oxygen, and nitrogen tend to be the molecules that life is based off of. But we also find phosphorus, sulfur, chlorine, sodium, magnesium, potassium, calcium, and a bunch of other elements in life. So you should know how to do a condensation reaction, how to make peptide bonds. We did that in organic chemistry, but also understand how complementary would be a necessity for the development of self-replicating molecules. Again, if we wanted information to go from one molecule to another, that information needs to be conveyed from one molecule to another, and you could do that by the molecules being complementary. Okay, so we're going to talk about section 1.2. So we'll close up that section and start talking about cellular architecture. So other things that were needed for life. So there's thought to be 7 distinguishing characteristics of living matter. And again, it depends on who you talk to, but this comes from Koshland, 7 Pillars of Life. It's thought that life needs a program, needs to be able to improvise. And what we're going to talk about in this section is compartmentation. So able to separate from the environment through membranes. Also needs ability to generate energy and process food metabolically to carry out chemical reactions, regeneration. So if there's an issue or an injury, you need to compensate for the wear and tear, you need to adapt to the environment. So most of the time the environment's changing, so you need some way to adapt, and then finally seclusion. So operate the processes in pathways and isolation from other things. So those are distinguishing characteristics. And like I said, we've talked about a couple of them, but we're going to talk about compartmentalization. So we're not quite sure how self-replicating macromolecules became encased in membranes. So if you're getting a common theme here, we don't know a lot about early life, but we do know that life now is encased in membranes, and there's advantages for that. Protection from the outside. You can actually keep your catalyst in the proper place. You can increase concentrations of reactions, so reactions that might not occur unless you have high concentrations. You can get those to occur within a cell. And then you can couple reactions. If this was just happening in a beaker, you could expect lots of side reactions or non-productive reactions. But within a cell, it's very organized. You can see a cross-section in a cartoon of an E. coli there. And within cells, we used to think that there was lots of empty space, but in reality, they are chock full of molecules. And so they compartmentalize and get the reactions that need to occur in the right space, right proximity for other reactions. Again, this is all stuff that we'll talk about in biochemistry. So if we look at life and where we're at now, we can kind of trace back and see similarities and differences between the different types of life on Earth. And it's thought that there was previously a earliest common ancestor back here somewhere, some type of self-replicating organism that through change was able to create what we think are now 3 different branches of this evolutionary tree. And so those are classified into bacteria, archaea, and eukarya. And so if you've had microbiology, you'll talk a lot more about this than we will. Again, we just want to understand that there are different types of life. They have very similar biomolecules, but they also have some very interesting differences between them. And so here is one way that we go to classify life, and that is between 2 types of cells, the prokaryotes and the eukaryotes. So the biggest thing for a eukaryotic cell is that it has a membrane-bound nucleus, where that's not found in prokaryotic cells. And if you find anything about life, you'll find that there's exceptions to a lot of the things listed here. So if we go back to this chart here, some of the things that we find, like these here, will also be, I can't find it, but you'll also find bacteria that have flagellum as well. And you'll find bacteria that have cell walls, and you'll also find some eukaryotes that have cell walls. And anyway, you'll find that this, instead of being a nice tree, is more like a bush, where some of the things that we like to classify overlap with other things. So it's not an easy division between these. There are some things that are what we would say are absolute between them, but just like we say cytoskeleton down here, in eukaryotic cells it's present, and in prokaryotic cells it may be absent, which means that some prokaryotic cells have cytoskeletons. And sometimes the differences are simply size. So large eukaryotes tend to have larger ribosomes, where prokaryotic cells tend to have smaller, and also eukaryotes tend to be larger, and prokaryotic cells tend to be smaller. But again, prokaryotes, you can find some that are 10 micrometers large, and you can find some eukaryotes that are 10 micrometers. So again, some of these are not absolutes, and it gets a little bit more complex when you start looking at the breadth of life. So if we start looking at just prokaryotic cells, so all modern organisms are based on the same morphological unit, which we refer to as the cell, which is just this membrane-bound compartmentalization of the biomolecular reactions. As I mentioned, there's two major classifications: eukaryotes, which have, in general, the biggest difference is a membrane-enclosed nucleus encapsulating the DNA, and the prokaryotes, which lack this nucleus. And the prokaryotes comprise various types of bacteria. They tend to be have more simple structures, and almost all are unicellular, although they can form filaments and colonies and films of independent cells that kind of work together, not quite as an organismal, but more like a tissue type level where they all are benefiting each other. Okay, and then if we look at a eukaryotic cell, we'll see that there's much more complexity there. So we can see that an animal cell, a eukaryotic cell, is much more complex, actually has several membrane-bound organelles within the cell. And so it's almost like layers within layers within layers, kind of like an onion. So we've got lots of different layers here. So not only do we have the compartmentalization of a cell, we have compartmentalization of other organelles, other functional entities within the cell. And that actually brought up an interesting hypothesis that it was proposed in the early 20th century and later refined by Lynn Margulis in the 1960s and 70s and even into the 80s, that eukaryotic cells are actually descendants of symbiotic bacteria, meaning a bacteria that would live within another bacteria. And so mitochondria are actually descendants of oxygen-respiring bacteria. So these oxygen-respiring bacteria set up home inside another bacteria, and then eventually that host bacteria took advantage of its housemate and made it a permanent guest. And this is kind of interesting to think about. A lot of people actually now are starting to believe this, but you do have a different set of RNA for your ribosomes, or sorry, not your ribosomes, your mitochondria, mitochondrial RNA. And something that's interesting is that everyone has their mother's mitochondrial DNA. When the sperm are formed, they actually need to shed a lot of the organelles, and so it just is a small cell that's packed with DNA, and so it actually gets rid of most of its mitochondria, and then when it fertilizes the egg, it has the female's mitochondrial DNA in there. So you tend to have your mother's or your maternal mitochondrial RNA. So you do get one chromosome from your mother, one from your father, but you are all the energy powerhouse of the cell, the mitochondria. You get that DNA from your mother. Thought the chloroplasts in plants and other organisms that harvest light are thought to come from bacteria that would actually use these chloroplasts for energy. Sorry, it's distracting. They're working on the road outside. Just a terrible day to try to present. Anyway, so if you hear the beeping, I'm not backing up. I'm not in a dump truck. I'm just sitting in my office, listening to the beeps. Okay, so let's close that up. Let's call this a lecture. I promise in the future they'll be better, as long as I don't have as much trouble with technology. And if they quit the construction, you feel that? I'm even vibrating. Okay. Well, good luck with the first lecture. We will have some more for you tomorrow when we get to talk about thermodynamics. How exciting will that be?